The first award talk, and I say talk in quotations as you'll see in a moment, is the Raymond L. Lindeman Award for an outstanding paper by a young scientist. This annual award recognizes an outstanding paper dealing with the aquatic sciences. Nominated papers must be written in English. These are the requirements, of course, but the important part is this award is to honor an early career scientist no older than 35 years during the publication year. The paper has to be written in a peer-reviewed journal 2 years before the award year. For example, this year's awarded paper, was written in 2006. The nominee must be first author and so on. If you just look at this list of illustrious awardees, it's quite impressive. No less impressive is our awardee for 2008, Cornelia Wuchter, who will be up here in a moment to talk about her wonderful work on Archaeal nitrification in the ocean, published in PNAS in 2006. Cornelia did this work while she was at the Royal Netherlands Institute for Sea Research. The work, as you can see, if it's not too pixelated there, has a tremendously illustrious list of co-authors. And now Cornelia is not too far from here as the crow flies, and she's currently a postdoc at Woods Hole. This work is already having a broad and profound impact. As you all know, I'm sure nitrification is a key process in the marine nitrogen cycle, and until recently, much of that nitrification was thought to be done principally by bacteria. Her data from enrichment studies and field work in the Atlantic and North Sea indicate that the my glasses are the Archaeota may dominate the process. This paper has already been frequently cited and is having a large impact on microbiology, biogeochemistry, oceanography, and earth science. This is the citation, and with that I'd like to ask Cornelia to come up, and also for reasons that Georgia will explain, Georgia Klein also will be here coming up, and Sybil Seitzinger will present the award.
So, Cornelia, I'm honored to present this award to you, and the award reads the Raymond Lindeman Award for Outstanding Paper in Aquatic Sciences by a Young Scientist. The award to Cornelia Wuchter, thank you, for I practiced that too, for fundamental contributions to understanding archaeal nitrification in the ocean. Congratulations.
Thank you. So this is a very bizarre situation. For the first time since I know Cornelia, which is since 1993, when we started to study together, she lost her voice. She's literally speechless. I tried to stuff her with fisherman friends and other pills. It didn't work, so until 1 hour ago, I had no clue what Cornelia's working on. so we spent the last hour up in her room cutting her presentation, because I would really like that this award is adequately recognized and that she has something from it. So I'm going to talk a bit about what she was working on. And actually, I'm an ecologist. I'm working on aquatic invasive species and have no idea how to pronounce these words properly. So please bear with me and be patient when I try to pronounce Archaeal, whatever, properly. So, okay, so let's start. Are you okay? She's more nervous than I am, actually. She's freaking out because somebody else is presenting her results. It must be terrible. So, okay. Yeah, okay. So Cornelia, she got her award for the archaeal nitrification in the ocean. She found out that the archaea are the major ammonium oxidizers in our oceans. As you can see on this slide, this tree presents the archaea forming one of the 3 domains of life. And within this domain, we find 2 major phyla, the Crenarchaeota and the Euryarchaeota. Originally, they were thought to be extremophiles, but recently it was discovered that archaea also inhabit marine environments, and that the marine group 1 Crenarchaeota constitute 20% of the world's ocean's phytoplankton. Despite the fact that they are so abundant, little was known about the physiology of marine Crenarchaeota. Marine archaea inherit specific membrane lipids. Stable isotope and radiocarbon analysis done on these lipids suggested that they might utilize bicarbonate as their carbon source. However, other studies show that they take up amino acids, which suggest they are heterotroph. So the major questions are they autotroph, heterotroph, or mixotroph? Leading to the questions, what is the carbon source of marine Crenarchaeota and what is the energy source of marine Crenarchaeota? The problem was there was no pure culture of marine Crenarchaeota, so Cornelia decided to work with mesocosm experiments in the Netherlands. We're going to show you a couple of pictures of the Netherlands and the crime scene, the sampling location, where she took her samples while she was doing her PhD in the Netherlands. So the experimental setup included 2 tanks with 20 liters of North Sea surface water each. One served as a control, the other was enriched with 100 micromole carbon-13 bicarbonate, which is about 5% of the natural DIC pool. They were incubated in the dark at 15 degrees Celsius for about 7 days. The lipid analysis showed the presence of archaea membrane lipids in the mesocosm water next to the bacterial lipids. Sorry? Yeah? Okay. So the isotope analysis revealed that the typical bacterial fatty acids had hardly any incorporation of carbon-13, whereas the archaeal lipids were highly labeled, and that about 70% of the incorporated carbon-13 was detected in the archaeal lipids. Thus, at least some marine Crenarchaeota appeared to be autotroph. Cornelia started an experiment to analyze the archaeal membrane lipids and their adaptations to different temperatures. And when she analyzed the lipids, she detected an enormous increase in the 22 and 25 degree Celsius mesocosm tanks. So she became curious and did a catfish analysis with a probe specific for Crenarchaeota and found that about 70% of the DAPI-stainable cells were marine Crenarchaeota. She measured nutrients and determined cell abundances in these microcosm tanks. And we see here the incubation time on the x-axis, and the graph shows that the Crenarchaeal abundance increases while the ammonium concentration decreases and nitrite increases. The well-known proteobacteria involved in ammonium oxidation were below 1% of the total cell counts. So could it be that the marine Crenarchaeota are the key players in the oxidation of ammonia to nitrate? She analyzed the archaeal AMO-A gene abundance involved in an ammonium oxidation and found the perfect match between cell abundance and AMO-A gene abundance in the mesocosm tanks. This led to the conclusion that marine Crenarchaeota are the key players in ammonium oxidation in the tank experiments. So the next step was to study the importance of archaeal nitrification in the marine environment. The graphs show a 1-year time series of nutrient development and archaeal abundance in the ocean margin in the North Sea. What you can see is that the decrease of ammonia and the increase of nitrate correlates with the abundance of Crenarchaeota in the water. In the next step, she analyzed the AMO-A genes of the proteobacteria and the archaea present in the water. And she found that the proteobacteria AMO-A is 3 orders of magnitude lower than the archaeal AMO-A, suggesting that the Crenarchaea is the dominant group in the nitrification in the ocean margins. Next, she had a look at open ocean samples from the Atlantic, samples from the upper 1,000 meter of the water column. And again, they were analyzing AMO-A genes of the protobacteria and the archaea. Please note the logarithmic scale. Surprisingly, the archaea AMO-A gene was 2 to 3 magnitudes higher than the well-known AMO-A genes of the proteobacteria, which suggests that also in the open ocean marine Crenarchaeota are the dominant ammonium oxidizers. And on the last slide, Cornelia would like to thank her PhD supervisors and all people involved making her work and publishing possible and successful. Thank you very much for your patience and attention.
Would you be willing to take a few questions through an interpreter? Are there any questions for Cornelia? I mean, other than about her voice or obviously how Crenarchaeal or something. Questions? Sorry. Yes, over there.
Is that pretty general, do you think, around the oceans or more localized in one place?
Well, she's telling me in German. Thank you very much. Okay. So she said they analyzed the first 1,000 meters of the Atlantic.
Yeah, transects, and it's very abundant.
It's very abundant there.
in the
So
ocean. So it seems they're dominant players.
seems to be the dominant players.
Anyone else like to torture Cornelia?
So what role do the heterotrophic Crenarchaeota play in all this? Are they the same or are they different?
Okay, there's work done. I'll try it. I hope you understand me. Well, there's work done, for instance, by Gerhard Handel's group, and they actually can show that you also have kind of Crenarchaea who are taking up amino acids. So I would never postulate completely say that all marine Crenarchaea are autotroph or chemoautotrophs, but we think that they play an important role, at least in the upper 1,000 meters of the water column. And right now there is actually evidence that in low oxygen environments, which are key places for nitrification processes, that it really seems that the archaea are really play a dominant role. But I would right now, in that stage, never go as far as to say that all marine Crenarchaea are chemoautotrophs. That's, yeah, I wanted to stress that out yeah, in my talk. So, but yeah, there's a lot to do still, because my work is also based on DNA. So we really have to also analyze the RNA to actually really showing the active expression of the gene. And right now, a lot of groups are now starting to really measuring nitrification rates and coupling this actually with the activity of the Crenarchaea to see what is their role. But potentially with 3 orders of magnitude higher genes versus the bacterial genes, it's, it really seems they're a really dominant player in the nitrification processes, which was until then not really known. So.
Questions with a yes or no answer? Thank you so much.
Thank you.